On a Sunday of 31st October 2021, a surface water sampling and analysis was conducted by Group 3 at Sunapago, Muar. The function of this river is to store the surface runoff from the adjacent road and the water discharged from the surrounding, as well as control the water level to prevent flood. Along the river have oil palm plantation, factories and houses. Before going to the sampling field, every equipment needed for surface water sampling were organized in field equipment checklist. This is to ensure completeness in carrying out on-site test. At the sampling field, it is very important to observe the sampling location in order to find the suitable sampling point that is easy to access and safe from any hazards. In the sampling location, grab method by using rope and bucket was adopted due to limited access to the river, and sampling was conducted at the high bridge. Besides, composite sampling was adopted by taking water sample from 6 different points, and pour them together into one. pH of the water sample was tested immediately before it was transferred into bottle. There are 2 preserving methods were adopted. The first is putting the plastic bottle containing water sample in an ice box, keeping it at low temperature to prevent changes in growth of microorganisms before it is placed at the fridge. The second is adding hydrochloric acid into a glass bottle containing water sample, until its pH is below 2, to preserve the hardness. Initially, the total hardness test was conducted. The sample poured into a 100 ml graduated mixing cylinder. The mixing cylinder was inverted several times to mix before 1.0 ml of alkali solution was added. 10 ml of the solution poured into each of 3 sample cells. Push read to read the result in DR6000 UV vis spectrophotometer. Next, chemical oxygen demand COD test was conducted by pouring the water sample into 250 ml of beaker and transfer into vial containing 2 ml of deionized water. And then the vial was preheated to 150 degrees CDRB 200 reactor. Lastly, its COD was determined with Hatch DRB 4000 reactor. Furthermore, in alkalinity test, phenolphthalein, methyl orange and bromocresol green methyl red were used as the indicators to measure the capacity of water sample to neutralize a strong acid. 6 units of 300 ml BOD bottle were labeled. Each bottle partially filled with dilution water and sample size was added. There is 1 bottle were filled with distilled water completely as blank sample. The remaining BOD and were placed in the incubator at 20 degrees Celsius and incubate for 5 days. The initial dissolved oxygen and final dissolved oxygen were recorded. Then, total bacteria count was conducted with plate or pour method. The total suspended solid, TSS, total dissolved solid, volatile suspended solids determination test were conducted. Generally, a 50 ml of sample was passed through a prepared, pre-weighed filter paper. The filter was dried at oven at high temperature for a period of time. After drying, the filter is reweighed and the TSS, TDS and VSS were calculated. Based on the results obtained, water in Sunapago is classified in Class 3, according to Interim National Water Quality Standard.